Hello, and welcome to this lecture in clinical genomics. This is the first in a series of 10 lectures that have been prepared for the residents in pathology at Stanford University, but we've also wanted to make the lectures available to anyone else who would like to look at them and learn from them online. Mine is the first lecture in the series, and it's sort of an overview and introduction. The title is Measuring and Manipulating DNA and RNA, and my name is Scott Boyd. I'm an assistant professor in the department. The other lectures in this series, following this introductory one, are going to go into more detail into particular topic areas, some of them sort of more technology focused, like studies of microarrays and analysis of hybridization data, and another lecture that looks at high-throughput DNA sequencing technology and the way that may impact future medicine. Then the rest of the lectures are really looking at different disease or biological conditions where you can actually apply these methods, and we'll discuss the impact that the technologies are having on the understanding of these disorders. So for example, inherited genetic disorders is one lecture. There are a couple that are on cancer genomics, solid tumors and hematopoietic cancers. We have a lecture on pharmacogenomics and one on transplantation-relevant HLA genetics. And then, importantly, there's a lecture that's addressing the whole concept of personal genomics and the kind of regulatory, ethical, and commercial considerations that have come into that topic area. So in this introductory lecture, I'm going to try to make ties between the technological advances that have increased our understanding of DNA and RNA molecules and the discoveries that have been obtained using those as a way to bring us up to the present time and the terrific period of discovery that we're living in right now. So the first point is the historical overview, discovery of DNA and RNA. And how did we really get where we are today? In thinking about this topic, many people go back to the mid to late 19th century where chemists, in particular organic chemists, were beginning to make progress in studying the very complicated chemicals and substances that you can isolate from living things. And one person who made a lot of contributions in this area was Johann Friedrich Miescher, who was working with the famous chemist Hop Seyler in Tübingen in Germany. And he took it upon himself to study the biochemicals that were present in the cell nucleus. Now this was at a time where microscopes had been around for a while. There was understanding that the cell has this separate compartment, the nucleus. But what exactly was in there and what it was for, or what its function was fairly unclear at that point. And Miescher, he took a fairly pragmatic approach to obtain material to study and isolated pus-soaked bandages from local hospitals that were just loaded with leukocytes, so the white blood cells, probably primarily neutrophils, as they're called. And these are the cells that he chose to study. By sedimenting the nuclei and then extracting the components of them, he was able to isolate a substance that he called nuclein. And on doing elemental analysis of it, he found that, surprisingly, it was rich in phosphate, and it was an acidic molecule, and it was relatively low in sulfur. And these are features that were quite different from many of the other biochemical isolates you would get from cells, primarily because protein would make up the majority of the material you would get from other kinds of isolations. So this was a new substance, and interestingly, the methods that he used are actually not dissimilar from things that are still used in DNA preps today, where you can use alkaline extraction to isolate DNA from the nuclei and then precipitate it with acid pH subsequently. It works quite well to the present day, of course. The insight that Miescher had about this discovery, they were not extensive. I mean, he didn't immediately say that this must be the material of heredity, but he's recognized as the one who first really isolated the first kind of semi-purified nucleic acids and found that they were in the nucleus. Interesting, though, imaginative scientists, even at this relatively early time, caught glimpses of what was to become what were to become very important concepts later on. And then there's this letter in 1892 where Miescher wrote to his uncle with the concept that large molecules composed of small building blocks could encode the information of heredity, just as the words and concepts of all languages can find expression in 24 to 30 letters of the alphabet. And at this point, he didn't really know that DNA was made up of, a structure similar to what he described in this sort of theoretical vein here. But in fact, that turned out later to be the case. And if we look a few years ahead, by the early 1900s, the following features of nucleic acids were known, or were thought to be known, and that they had been found in all nucleated cells that had been evaluated, that their components included ribose sugar, phosphate, and 5 different kinds of nitrogenous bases: guanine, adenine, cytosine, thymine, and uracil. And those bases were actually characterized by Kossel, who was another student of Hopsaler, so that was a real hotbed of these early biochemistry/molecular biology discoveries. And at the time, though, there were still a lot of things that were unclear. And for example, it was thought that DNA might be the primary nucleic acid in animal cells, whereas RNA might be the major form of nucleic acid in fungi or plants. So there really wasn't yet the clear picture about the order of transmission of information in sequences. A lot of the other details of the building blocks that make up nucleic acids were discovered over a fairly longish stretch of time. So one of the features in this whole area is that at the time it was really thought that DNA and RNA were simple repeating polymers with a structural role and probably didn't have enough complexity to them to actually encode the hereditary information to confer such complicated traits as eye color and all the other features that seem to be able to be inherited by organisms. In terms of the increased understanding of what the building blocks in detail were of nucleic acids, this guy Phoebus Levine, who was the head of biochemistry at the Rockefeller Institute, he played an important role for better clarifying some of the aspects of the backbone of DNA and RNA. For example, he characterized the sugar ribose, which is the sugar that forms the part of the backbone of RNA. And 20 years later, he had characterized deoxyribose. And similar to the work that was done in the 19th century, a lot of this was very painstaking, real organic chemistry, and was more difficult things to figure out in great detail exactly which atoms were bonded to which in fairly large molecules for the time. This Phoebus Levine is also known for proposing what was called the tetranucleotide structure for DNA. And it's fascinating to look at this because basically the pieces are essentially correct. You have ribose sugars joined to phosphates connecting them, so that's the ribose, in this case deoxyribose phosphate backbone. And he has the bases attached to the 1' position as they would be. But here there isn't the recognition that the DNA could be a long polymer. It was basically these little rings of 4 nucleotides each. And so that if you had that sort of a framework in mind, you wouldn't be expecting that the DNA or RNA would actually be the molecule of heredity. However, other experiments soon began to point in that direction. There was a researcher, Frederick Griffith, who was working in Britain at the time in the Ministry of Health, and he made some very interesting findings related to the transmission of genetic information, seemingly, or the ability to at least transfer traits between different bacteria. And he studied pneumococci, so strep pneumonia, and he was studying different strains. And there were some strains that were rough in the morphology of their colonies. These were non-virulent. He studied other strains that were smooth with their capsule, so they made a lot of extracellular material. And these were the virulent bacteria. And if you injected the rough strain bacteria into a mouse, the mouse could fight off the bacteria and survive. If you injected the virulent smooth strain, however, the mouse would die from the infection. And Griffith did this experiment where he found that if he took the smooth strain, the virulent one, and killed it with heat, then injected it, the mouse was able to survive. So the bacteria, the virulent bacteria had to be alive to kill the mouse. But then, very interestingly, he took the rough strain, which by itself wouldn't kill the mouse, and added the heat-killed smooth strain. So the combination of these things together, injected into the mouse, suddenly restored the virulence. The mice died under this treatment. And this was a big surprise. It gave the impression that there was a transforming principle, as it was called, that could pass the virulence trait from the heat-killed smooth bacterial strains to the live non-virulent ones. Now, what the nature of that substance was yet to be discovered. However, people fairly soon after that began to attack that question, and one of the leaders in this area was Oswald Avery. He was working primarily at the Rockefeller Institute after having worked in bacteriology, including studying topics relevant to food preparation like yogurt. And Oswald did these really nice experiments that I think are classic early molecular biology, verging on biochemistry, to try to figure out what was the component in the Griffith experiment that was able to pass the information that caused the rough pneumococcal strain to become smooth and virulent, because it didn't require live bacteria. It was some component of the cells. So what he did is he and his collaborators took these smooth pneumococci, the virulent ones. They isolated a cellular extract which had, of course, protein, RNA, DNA in it, as well as lipids and other things. But then he took that extract and he treated it with several kinds of destructive treatments to knock out each of those different kinds of components, and to see, of those that were left, which of them could actually confer the trait of being virulent or of creating the smooth colony morphology. And what he found is that if he didn't use any extract and he added the stuff to rough pneumococci, the non-virulent ones, there was no change. If he added the extract without any treatment, then, as you would expect from the Griffith experiment, you would actually get smooth colony formation, so the virulence had been conferred onto the live non-virulent pneumococci by something in the cell extract. Now, if you treated the extract with RNAs to destroy RNA, there was no change. You could actually still get the virulence being transmitted. If you treated it with proteases that would destroy many proteins, again, the virulence was still able to be transmitted from the cell extract of the virulent cells to the non-virulent ones. But when he treated with DNAs, suddenly the ability to confer the trait actually decreased a lot, and he got primarily rough colonies. So this was reasonably good evidence that DNA might actually be the key thing that was able to pass this trait from the one bacterial strain to the other. And so that pointed to DNA as potentially really having a bigger role in inherited information or hereditary information than had been thought before. And I think at the time there was still a lot of doubt as to whether things that applied to bacteria, or especially to bacteriophages, would necessarily apply to all the other more, the larger forms of life in terms of organisms like humans and others. But it at least pointed in the direction that DNA, under these circumstances, could actually confer hereditary information. So some of the other key pieces of the puzzle began to fall into place in the subsequent years. Erwin Chargaff, working at Columbia University, he had been studying the chemistry and features of DNA in great detail, and came up with what were called Chargaff's rules, which were, he found that if you took a particular DNA isolate from a given organism, you would actually find that the amount of A, or adenine, that you isolated from that DNA was about the same as the amount of thymine. And similarly, the amount of guanine was about the same as the amount of cytosine. And actually, if you looked in different kinds of organisms, plants, different bacteria, mammals, and other animals, what you find is that the overall proportions could vary quite a bit. Some organisms had more A and T compared to G and C. Others had different proportions. But that this matching rule of A and T being similar, G and C being similar, pretty much held up. And that was for DNA. And at the time, he was using the exciting new technological methods of paper chromatography and UV spectrophotometry, which, if you compare it to the methods that others had used previously, were big advances and really allowed you to separate longer molecules, more closely related, more chemically similar molecules. At the time, there were still many things that were unclear about DNA. It was known that you could form fibers of it, and if you do just a very simple DNA isolation, you can actually spool the DNA out in what looks like strands or fibers. But it wasn't really clear. Was it truly a linear molecule? Did it have some other structure that we didn't know about? Was it branched? And if that sounds strange, I mean, subsequently it's been learned that there are contexts where, certainly in the case of RNA, you can have branched structures, like in the lariat structures that are excised during splicing of introns, for example. There were other steps along the way that led to the current understanding of DNA and its function, and a lot of them, as in the case of the bacterial experiments that I described, made use of simpler genetic systems that allowed you to do well-controlled experiments and study things reduced down to a sort of very isolated subset of the phenomenon that you were looking at. So in this context, the phage group of scientists that became organized around Cold Spring Harbor, particularly with Max Delbruck and Salvatore Luria, made big advances here. And Delbruck had come from a physics background, and I had thought it would be interesting to have a look at what was going on in biology and study exactly how it is that these phage, or what were subsequently found to be viruses that have as their host bacteria, how they reproduce, because there was some earlier knowledge that he played some role in, that in contrast to the division of cells like bacteria, which follow a sort of exponential growth pattern, bacteriophage seem to reproduce in a manner where you got not much happening, and then all of a sudden you get a big explosion of many progeny resulting. So it seemed like there was something different about the way that bacteriophage reproduced themselves and destroyed the bacterial host that they grew in. And it seemed like a relatively feasible, small, simple system to study. The bacteriophage, don't have that many protein components. They had nucleic acid associated with them, but it wasn't huge in its length, for example. And this was appealing to those who were used to thinking in terms of reductive physical systems and had worked on them previously. And this is also the same group of investigators where James Watson, for one, had some training as well. So some of the experiments that I think contributed to the importance of bacteriophage and in modern molecular biology understanding, and that also helped to reinforce the idea that DNA really probably was the molecule that can confer genetic traits or genetic information, was this Hershey-Chase blender experiment. And this is one where these researchers, they made preparations of bacteriophage in which they either radioactively labeled the DNA inside a DNA phage of this kind, the T2 phage, which is one of the T-even bacteriophages, or else they made a version of the phage where they had labeled the protein with S35 label on it, so sulfur with the radioactive 35 isolate or isotope. And what they found is that you could allow the bacteriophage to begin to infect bacteria, so they settle on the bacterial surface, they inject through the cell envelope their nucleic acid, and what they then found is if you use a wearing blender or a high-speed blender to shear the bacteriophages off the surface of the bacteria, what you found is that under conditions where infection had definitely happened and the infected bacteria began to produce more bacteriophage, it was the labeled nucleic acid that had been transmitted. At this point, you could have still made the hypothesis maybe there's some small component of the total proteins of the phage that were the actual carrier of the genetic information allowing the phage to reproduce itself. But it was found that in the case of the protein-labeled phage, that there really wasn't anything inside the bacteria, and in this case, the infection went along fine. Those bacteria began to produce new bacteriophages, but clearly the labeled protein hadn't been necessary to get into the bacteria to cause that to happen. So this gave a little bit more support to the idea that DNA might be the key molecule transmitting genetic information. Developing in parallel with the things that I just described was the whole area of structural biology. And some of the early pioneers in this whole area were William Lawrence Bragg and his father William Henry Bragg. Henry Bragg, I think, essentially was the originator of X-ray crystallography, and it was applied most enthusiastically by his son Henry Bragg in the early days. And they really showed that if you use X-rays to irradiate crystals of any given substance, you can use the refraction patterns from the crystal to identify what the structure was or what the positions of each of the atoms in those molecules were. And so that was extremely powerful new tool, because it allowed you to use wavelengths of radiation that were short enough to really be isolating the information at an atomic level, and to study in great detail the structure of compounds that otherwise may have been very challenging to study by traditional chemical techniques, and that certainly no microscope of the period would have been able to help you evaluate. Other people who were very important in this area were J.D. Bernal and Dorothy Hodgkin, who were Hodgkin studied with Bernal, and between the two of them, they basically tested to see whether from apparently very complicated molecules like the protein enzyme pepsin, that you could actually crystallize those things, and from those crystals you could get good diffraction patterns similar to what had been done with other kinds of organic molecules by the Braggs and others. So this really was the first hint that you could actually use these techniques to potentially figure out what complicated molecules like proteins and nucleic acids looked like. Around that time, a researcher at the University of Leeds named William Asbury was also doing studies along the same vein, and he showed that if you obtain diffraction, so X-ray diffraction patterns from wool, which is primarily made up of the protein keratin fibers, that you could actually get a different pattern of diffraction whether or not the wool was stretched or unstretched. And in later years, it was realized that this actually corresponds to two of the major kinds of secondary structures that are the sort of second-order building blocks of protein final structures. The unstretched wool had a pattern that turned out to be something called the alpha helix, and the stretched wool had a beta pattern, which turned out to be what's now called the pleated sheet kind of secondary structure motif. And he also, this Asbury, generated some early diffraction patterns of DNA fibers that gave some hope that you could actually try to learn something structural about the DNA by using these X-ray crystallography methods. And someone else who took a big interest in this whole area and had been applying X-ray crystallography to study smaller molecules and crystals and various things was Linus Pauling. And this was after his formidable work in better explaining chemical bonds and the electronic structure of atoms, and how that might explain which atoms were bonded to each other in more complicated molecules. So Linus Pauling began a program of more biologically focused research and began to apply crystallography to various biological macromolecules. And in relatively short order, he was able to elucidate the alpha helix crystal structure and sort of draw or create models of it that fit very well with the experimental data, as well as the pleated sheet structures that were found in proteins. And he soon began to work on DNA as well. Around the same time, researchers at King's College London, particularly Rosalind Franklin, who was working under the overall group of Maurice Wilkins within that institution, Rosalind Franklin had been obtaining high-quality DNA diffraction photos from fibers of DNA, and had been generating some of the background data that you would want to acquire if you were going to try to figure out what the structure of DNA was. Now, at this time, this is when James Watson and Francis Crick were beginning to think about the structure of DNA and how you could begin to figure out what it might be. And Watson made a visit to King's College and had some discussions there, in the course of which I guess Maurice Wilkins showed some of Rosalind Franklin's data to Watson, and he saw, for example, this famous photograph 51, I think it's called, of the diffraction pattern from DNA, and he could see right away at that point, Watson wasn't very sophisticated in terms of understanding the correlation between the diffraction patterns and what the underlying crystal structures might be. But he, by his own account, recognized that the X said right away it should be a helix. And I guess over dinner, apparently Maurice Wilkins also described some other features that you could infer from the pattern. For example, that I think there was a missing pattern of bands here that could indicate that this is where the sort of crossover of the two strands, if you were looking at a particular angle, would occur. And that gave the hint that there might be something like major and minor grooves. And it also allowed some parameters for the spacing of the elements within the helical structure to be identified. And so this gave a lot of insight right away, and was further mulled over by Watson and Crick once he got back to Cambridge. This helped a lot in the development of the double helix model of DNA structure that Watson and Crick proposed in their 1953 paper. And this was a total bombshell of a discovery, because it immediately suggested ways that the genetic information could be stored and copied, in particular by tying the Chargaff rules of adenine and thymine being present in similar proportions and guanine and cytosine being present in similar proportions, with the idea of a double helical structure, and in this case one where, because of other features of the crystal structure pattern, they could tell that the bases should be on the inside of the helix and the ribose and phosphate backbone should be on the outside. It indicated that if you knew the sequence of bases from one strand, you could probably infer what the pattern should be on the other strand, so that when a cell is dividing, all it needed to keep was one copy of any particular DNA molecule, or one strand, and it could reconstruct the other strand. So a very simple and elegant method for storing and copying the information of the DNA. And so that had a huge impact, clearly, going forward. Subsequent research focused on the question of, well, how do you actually get from what may be the stored information in the DNA to the final sequence of the proteins that actually do a lot of the work in the cell and form useful molecules like enzymes and structural components. I won't go into a lot of detail about this, but there were definite hints that DNA was able to transmit its information to RNA, and that RNA would then code for the protein sequences. But there were a few steps along the way. And to carry on the thread of the bacteriophage studies, another major contributor in this area was Seymour Benzer, working at Purdue University. And he used a different kind of DNA bacteriophage, the T4 phage, as his preferred system, where he was hoping to be able to map the genetic elements in the bacteriophage by using recombinations between mutants. So he was able to study mutant bacteriophage, for example, that by themselves were unable to infect a particular strain of E. coli. But what he found is that if he took two different kinds of mutants that subsequently he was able to figure out had mutations in different parts of the bacteriophage DNA, and have both of them infect a bacterial cell, that sometimes you would get a crossover event where you would actually have a breakage and then a rejoining, so that the two different strain DNAs swapped halves of their overall chromosome. When that happened, the result was you got one piece of DNA that had both mutations on it. The other piece was wild type, and now that wild type bacteriophage could actually infect the E. coli productively. So this is something that you could then measure by the ability to form plaques on bacterial colony lawns, or bacterial lawns, I should say. And it really allowed very precise mapping of the positions of particular mutations with regard to each other, because the further away two mutations were, the easier it is to have a crossover event happen between the two of them and cause them to become joined together, leaving a wild-type chromosome. And in addition, you could actually characterize different kinds of mutations. So instead of a point mutation like these two that are depicted here, you could have a deletion mutation, which you would be unable to recombine with, or would basically not be able to be rescued by swapping the same genetic element from a different phage chromosome. So you could distinguish between deletions and point mutations. and come up with things to do with the spacing between different lesions. And so that actually allowed mapping of some mutations to a single nucleotide level within the phage DNA. And these kind of tools became very helpful later on when Francis Crick and Sidney Brenner were working together after the double helix discovery and so on. They were trying to figure out, well, if there is a code in the DNA, and then potentially it was realized the RNA, that was encoding for the amino acids and proteins, what form would that code take? Some calculations, I think first proposed by George Gamow, another researcher with a physics background, had indicated that to encode the 20 plus amino acids that were present in humans at least, you would need to have at least 3 bases, because there are 4 possibilities for each position. So if you had only 2 bases, you could only encode 16 different things. So they sort of figured out that there would be a triplet code, and by using this sort of phage recombination and mapping strategies, they were able to figure out that it was a triplet codon. In other words, if you removed 3 bases at a time, you could still get most of the proteins produced, with relatively normal function, but that if you lost less than a multiple of 3 bases, you would then cause a frame shift, and that would totally disrupt the sequence of the protein following. So these bacteriophage tools were really helpful in figuring out some of those features about what the strategy for coding amino acid assembly was that was encoded by the DNA. And again, this is a very condensed description, so apologies to any of those who may have been involved in this. The final details about, well, what is the code for translating proteins from the information that's passed by DNA through RNA? And Nirenberg and Matai came up with a really nice way to explore this question. What they did is they created synthetic homopolymer RNA, so in other words, RNA that had only one base, poly-U, for example, polyurine DNA, or RNA, I should say. And they had a cell-free translation system, so basically bacterial extracts that were able to translate RNA into protein for a certain amount of time. And what they did is then put these synthetic poly-U and other homopolymer RNAs into the system, and they just saw what kind of labeled protein they had produced. And they were able to tell that by doing 20 different versions of the experiment with a different amino acid radioactively labeled each time. And so what they found is that if the RNA was poly-U, so it had a series of U's, that the protein that they got out was phenylalanine only, so polyphenylalanine. And therefore the codon for phenylalanine was UUU. Similarly, an experiment with poly-A gave a polylysine protein, so therefore triplet A was the codon for lysine. Poly-C ended up being the codon for, or triplet C became the codon for proline. And that was good as far as it went, but then clearly there were still a lot of other codons to figure out. And so several other researchers, including Choa and Khorana, and several others who had figured out how to be able to synthesize nucleic acids to specifications where you could have a particular order of bases. That was very useful. And in addition, systems where you didn't actually have to be producing a long protein to characterize, but could actually just determine, using only 3 bases of RNA, determine which tRNA loaded with the amino acid in question would be bound to that in the ribosome. So with Phil Leader and others, Nirenberg was able to basically continue to map out the rest of the codons just by using the little trinucleotide RNAs representing each of the codons and seeing what kind of ribosomes were able to associate with those and what amino acid was also associated with those ribosomes. So it was a laborious process, but eventually the codon code for each of the 20 naturally occurring amino acids in humans was figured out that way, or in most other organisms. So that ends the historical part of the talk. By this point, some of the broad outlines about the structure of DNA. I didn't talk too much about the discovery of RNA, but there was a lot of work that had to go on there distinguishing ribosomal RNAs from tRNA, from the messenger RNA that's the real transcript of the DNA information. But what this did is it opened the door to a lot of things that were immediately very useful for medical discovery. And now I'm going to go through some of the technological advances in the years since then, which have led directly to amazing discoveries about how cells and organisms work. So this first area is hybridization-based technologies. And so this stems from what I described, something that was made as a very exciting possibility when the double helix DNA structure was proposed, which is that if you had one strand of a nucleic acid, of DNA in this case, and you had an unknown pool, you could actually try to get the two to hybridize together and basically detect whether or not the complementary strand to the one that you had a label on was actually present in the other pool that you were examining. And so, for example, one can do this under very stringent conditions where there's high temperature denaturing agents present. In that case, only target sequences that match almost exactly will hybridize properly. You could also do it under lower stringency conditions with lower temperature and fewer or no denaturing agents at all present. And in that case, you get a more lenient hybridization where molecules that are not identical but are somewhat related to the one that you're using as the probe in hybridization can be detected. And each of these conditions can be useful under different circumstances. And the overall theme that if you have something that you can interrogate an unknown pool of nucleic acids has been a very helpful strategy in lots of different cases, as we'll see. So some of the very appealing discoveries that were made using these kind of methods include things like the discovery of the human RAS oncogene. And this is a case where in the laboratory of Bob Weinberg at MIT, they did a nice experiment where they isolated DNA fragments from human tumor cells. The idea being, well, perhaps human tumors have altered genes in them or some sort of genetic changes that you could actually then use to confer the trait of cancer cell behavior onto other cells. And so these human DNA fragments were put into mouse fibroblast cultures, these NIH3D3 cells. And these cells, the 3D3 cells by themselves, they don't act in a very vigorously transformed manner. They tend to form colonies, but they don't continue to grow hugely, and they have other features that are not fully like cancer cells. But if you put the DNA from the human tumors into them, what you found is that you would get colonies of cells that were actually growing and had more traits that were similar to human cancer cells. And the idea was, well, maybe these are ones that have taken up cancer-related genes from the human DNA that we put into them. So then the DNA from these cells was isolated, was broken into manageable sized pieces using other tools that, again, were adapted from bacteriophage understanding. And you then try to figure out, well, of all this DNA that came from what are mainly still mouse cells, can we find any human DNA in the mix? And the way that that was studied was, it turned out that there are some sequences that are present scattered throughout the human genome, repetitive sequences that are now called allorepeats, that are characteristic of humans but are not present in mice. And so what you could do is you could run the mouse cell DNA on a gel, and you could actually look to see whether you could detect the presence of any human DNA in the mix. And so basically you were able to identify which of the bacteriophage that you had isolated the DNA inside contained human DNA. So this is an experimental method for saying, well, of the DNA that was in these mouse cells that are now growing in a more cancer-like fashion, well, what human DNA did they actually get? Voila, now you have this phage that has human DNA in it, and you're able to say, well, this could be a candidate for a human cancer gene that's been transferred into the mouse cell line. So when the DNA from that phage was then studied, it turned out that it actually contained a human gene, which has been since called RAS, that actually had similarity to other genes that had been found in different kinds of viruses that were able to cause cancer directly by infecting cells. And so this was the first discovery of what's been called an oncogene, or a gene that when modified or when mutated can be a driver for the causing of human cancers. A second, well, and just to make the point, this relied on the hybridization technology, which is the basis for doing anything like a southern blot, which is the kind of thing we have depicted here, where you're testing for the presence of a particular kind of sequence that you know something about using a probe that you've labeled. A second major area that made use of hybridization for a major biomedical discovery was the discovery of VDJ rearrangements, or the way that the antibody and T-cell receptor genes are produced in humans and in other mammals and other vertebrates. In this case, the question was, well, it had been a mystery for a while. Humans and other animals were known to make lots of different kind of antibodies. And those antibodies had different binding specificities, and there seemed to be just tons of different kinds. So then that raised the question, well, how on earth does the genome code for all those different kind of proteins? Is there a different gene for every kind of antibody? It seemed from the number of antibodies like that might not be the most likely explanation, but it was hard to know what other possibilities there might be. And there were these very influential experiments that were done by Susumu Tonegawa and his lab members, where he basically isolated DNA from both mouse embryos, so the whole organism, and also from cell lines that were from a malignant B cell kind of lineage, or plasma cell in this case, which is this 321 cell line. And the idea here was he was able to obtain RNA from another kind of B cell derived tumor, or a plasma cell line basically, and make a radioactively labeled version of that. So he had an RNA probe, and there was a particular way that, but in the process of radio labeling that RNA, you would actually occasionally get cleavage of it. So you could actually have a probe that would recognize only the 3' half of the antibody gene, or you could have the more intact version of the probe, which would label the whole antibody gene. And now this is at a time where one was just curious, what is the nature of these antibody genes at all? And what you'll see here is this is a graphical representation of the results from running a gel. So the high molecular weight or long fragments are up at this end, and the short fragments or low molecular weight things are down here. And what Tonegawa and colleagues identified was that if you look at the embryo-derived DNA, you found that there was one peak here, which you identified with both the 3' half of the RNA probe as well as the whole RNA probe. And there was a second peak here, which you only identified with the whole probe. And then way downstream at these smaller fragments, there was a whole different peak that came from the plasma cell or the myeloma cell line, in which case you'll notice here that both the 3' half and the whole probe identify the same peak. So the result that was inferred from these data are that in the plasma cells, you actually have a rearrangement so that the 3' end and the 5' end of the gene are brought together so that they're in the same piece of DNA. And I should mention that the DNA, before being put into this experiment, was cut up with restriction endonucleases, or enzymes that specifically cleave the DNA in particular positions, and those had only recently been described and been able to be isolated. So what this is saying is that the same fragment actually contained the 3' and the 5' end of the antibody genes here, whereas it looked like in the embryo of the mouse there was actually a separation between these two things, and it was a pretty considerable separation. So that gave rise to the idea that what's happening when you are making an antibody gene is that you rearrange the genomic DNA in part of your genome. And indeed, that's turned out to be the case, and now we know that actually every antibody is made by combinatorial rearrangement of lots of different gene segments. In the case of the heavy chain of the antibodies, there's a V, D, and J segment that get rearranged together. And this is, as far as is known in humans, this is the only circumstance where you really cut out part of the genome in a particular cell type in order to make a functional protein. And so in this case, it's a way of generating diversity as well, because you can combine many different gene segments and give rise to a great diversity of joined fused products. But this early experimentation, it relied on that same principle of hybridization. So once they had run their gel, what Tanagawa and his colleagues did was they cut out little slices of the gel, and in each case they tested to see whether they could get hybridization with the two probes that they were testing. They measured the amount of hybridized product that they got, and that was how this discovery was done. So this gave evidence for somatic rearrangement of the genes, or the gene segments that code for antibodies, and it was later shown that the same thing was true for T-cell receptors. Coming up closer to the present, although it's surprising to realize that it's almost 20 years ago now, a third very useful way to apply hybridization technology was in the miniaturized version, which became called microarray hybridization. So in this case, what you've done is you've spotted using tiny little pins or other methods of depositing DNA on a solid surface. You've deposited DNA for different genes or different other sequences of interest, and you can then take a labeled version of the nucleic acids, for example, the messenger RNA produced by a cell, hybridize it with these spots that are on the array, wash away the stuff that's unbound, and detect how much of your labeled sequence is present at each position on the array. So with this methodology, which was pioneered by Pat Brown and other colleagues at Stanford, what it did is it really opened the ability to measure many, many genes at the same time from a single cell sample, for example, or from small cell numbers, or just in general any kind of cellular population, but to make many measurements in parallel. And that's resulted in a whole area of kind of big data biology, where you're measuring many, many different events from living things all at the same time. And this method of measuring mRNA levels is actually one that's led to a variety of insights into cancer cells and other diseases, and it's had a big impact. Another methodology related to this is FISH, or fluorescence in situ hybridization. And this is something that's used in general usually on the DNA when it's still bounded by the nucleus of a cell. So you make the nuclei permeable, and then you put in labeled probes that will bind to a particular region of a given chromosome. What this methodology lets you do is detect whether you have rearrangements of the DNA in the chromosomes. And so, for example, in this case here, this is of a particular kind of leukemia called acute promyelocytic leukemia, and the two probes in red and green, those are on different chromosomes. One is on chromosome 15, and one is on chromosome 17. And in a normal cell, you'd expect to see 2 green dots, 2 red dots. But in this cancer, what you have is a translocation that fuses half of, or a portion of chromosome 15 to a portion of chromosome 17. And similarly, the other two halves are joined together as well. So you get these fused spots that show that there's been a translocation that occurred. And this approach can be used to look for deletions and amplifications of particular chromosomal regions as well. So it's been very useful in cancer diagnosis, for example. Okay, the next major area I'm just going to talk about is enzymes and molecular cloning. So this is something I've already touched on a little bit, the development of restriction endonucleases, which were enzymes that it was found could cut the DNA at very particular kinds of sequences, often palindromes, but not always, and would allow you to basically cut DNA not exactly where you wanted to, but at least in reproducible ways. It was also identified that there were other enzymes called ligases that could then join together DNA that had been cut apart. And so this really was the opening point for being able to manipulate DNA a lot more specifically than had been done before. As I mentioned, the restriction endonucleases were key to the ability to do things like southern blot or the kind of hybridization experiments that Tanagawa did in his experiments. So the trio of names that often come up in this whole area are Cohen, Boyer, and Berg, because they were really the pioneers in terms of recombinant DNA technology. And those are experiments where you cut DNA in a particular place or places, and you put in other piece of DNA, which in this case is labeled as the insert, then you fuse the two of them together. Now, in the early days of this, and the cutting is done by restriction in nucleases. You separately cut the DNA from which the insert comes. You cut the DNA that you're going to put it into, and then you ligate the two of them together with DNA ligase. And I'm not going into details here that there are different kinds of blunt versus sticky ends and ways that the restriction end nucleases can leave the site where they cleave. But the principle is that you can fuse different pieces of DNA together. And if you were to ask somebody around this period of time, well, what happens if you put a human gene into a bacterial piece of DNA and put it in bacteria? Will it be expressed, or will it be able to be even carried successfully there? Many people would probably say no, or at least that would have been regarded as maybe a long shot. But it turned out that in fact that is quite feasible, and then there was a lot of fear raised about, well, what are the implications of that? And some of those questions haven't been fully resolved to this day in terms of if you can manipulate DNA, you can potentially modify pathogens to make them more dangerous and have other various potentially malicious uses of this kind of technology. In any case, the methods of cutting DNA, and in particular of using these small circular pieces of DNA called plasmids that many bacteria carry, that are able to copy themselves within the bacterial cell when the bacterial cell divides and propagate themselves, they became very useful vectors for carrying other pieces of DNA that you could then incorporate into them. And often these plasmids carry genes like antibiotic resistance genes. Those can make it easier to tell when you've got a bacteria that's got the desired plasmid in it, because you can select for the presence of the antibiotic resistance gene, and you know that you've incorporated your desired insert into that plasmid. So there were several experiments leading up to this one that I described here, but the most striking example might have been when these guys cloned a piece of African clawed frog, or Xenopus ribosomal RNA. A fragment of that, of the DNA of it, was cloned into an E. coli tetracycline resistance plasmid and was able to be propagated in the bacteria. And as you can imagine, this ability to put in foreign pieces of DNA and have them be copied by the bacteria, and even expressed as protein by the bacteria if you set things up properly, that has given rise to the whole biotech industry of producing proteins, potentially human proteins, in systems where you can actually crank out a ton of the desired protein product. And later versions of this were able to do the same thing with mammalian cells using different kinds of vectors, but basically the same kinds of strategies. So from that, and particularly Boyer had a big role in this as one of the co-founders of Genentech, which led eventually to the company making the generous gift of a building to UCSF, where Boyer had a working connection previously. This led to the rise of this amazing biotech industry, which now is the source of lots of therapeutic proteins that help people with various different health conditions. Erythropoietin being one of the early examples, but recombinant insulin and a whole host of other protein therapeutics. Okay, the next thing I'm going to talk about is the PCR reaction, or polymerase chain reaction, and that's something that was discovered by Kary Mullis, who was working at the Cetus company at the time. This is really, I just talked about the cloning of DNA into plasmids and being able to use bacteria to copy it. That was extremely useful. PCR became an even more flexible way of amplifying a piece of DNA that you're interested in. And the way it works is the following. Mullis figured out that if you took a DNA duplex, so 5' to 3' in one direction, 5' to 3', the complementary strand, you heat it up to denaturate, the strands would separate. If you now allow those separated strands to anneal by lowering the temperature, with a small little piece of DNA that hybridizes to a particular portion of the denatured strands, and then you add in a DNA polymerase and deoxyribonucleotide triphosphates and the other conditions of salt and magnesium to have the reaction conditions appropriate, you could have, using this little primer as the starting point, you can make a copy of each of those strands respectively. And now that you have these new duplexes, part is the new strand that you've just made, part is the original strand that came in, you denature those again, and again hybridize with with primers. And what you'll notice now is, again, like in the first cycle, you get the primer hybridizing, you get the new strand being made using the original strand as a template. But now the copy that you made in the last round is also able to act as the template for a new round. So now what you'll notice is that a new primer is hybridized to something that was actually synthesized in the previous round. And what you can see is if you carry on this process of denaturation, hybridization of the primers, and extension with the polymerase, you could actually make what will gradually become an exponentially expanding number of these small products that are bounded by the primer sequences that you picked to begin with. So if you do that with enough cycles, you can amplify from a single DNA duplex or even just one of the strands of the DNA. You can actually make millions and more copies. So this was a very useful technology for provided you knew where you were going to have the primers for amplifying a sequence of interest. And some other innovations that were clever came along. Like initially, when this was done, the temperature that you used to denature the DNA strands would be enough to kill off the polymerase enzyme from the previous cycle. So you'd have to keep adding fresh polymerase in every extension cycle. But then it was figured out that you could actually use heat-resistant polymerases that were isolated from bacteria that naturally live in hot springs to stay in the reaction, and they were not bothered at all by the denaturation temperature. So you can actually do this PCR reaction without having to reopen the tube and put in more enzyme each time you want to extend the new DNA strands. So PCR has really become a workhorse of modern DNA manipulation and is built into almost every kind of, or at least many future strategies that were used. And just to highlight a couple examples, PCR was used to highlight several very interesting biological features of certain diseases. One of them was Hodgkin lymphoma. It's a lymphoma that's found both in children and adults. It has an unusual biology compared to some other kinds of lymphomas where the patient will have a hugely swollen lymph node, and the lymph node will be packed almost entirely with the malignant B cells or T cells potentially. In the case of Hodgkin lymphoma, there were what looked like very large abnormal tumor cells present in the tissue section, but they were surrounded by inflammatory cells that seemed actually not to be necessarily part of the malignant process. They may have been cell populations that were attracted in by the small number of these large tumor cells sitting in the middle of the lymph node. And PCR was used to evaluate what the nature of those large lymphoma cells in the Hodgkin lymphoma are. Because prior to that point, it was a little uncertain. They could be things related to macrophages or histiocytes. What exactly their lineage was, from what kind of cell they originally derived, was still unclear. But in a nice paper, a bunch of papers around this time, PCR of the nucleic acid from single cells, so the single tumor cells were able to be microdissected or isolated from these lymphomas. And then PCR amplifications were done to see if they could amplify rearranged immunoglobulin heavy chain gene rearrangements. And that's a key thing, because only in B cells would you have these IgH rearrangements typically. And that's, it is true that in small numbers of other kinds of malignancies, these things have occasionally been reported, but by and large, this would be taken as a very strong marker that the tumor cells were of B cell origin. And indeed, when the experiment was done, it was found that the Reed-Sternberg cells, or these large tumor cells present in the Hodgkin lymphoma, they did seem to have rearranged IgH, and you could really only measure that using this single cell PCR. So the great sensitivity of the PCR method enabled this bit of biology to be discovered, and subsequently there's been a lot of other evidence supporting the idea that Hodgkin lymphoma comes from a B cell origin, and that's something that I think was a key insight that wouldn't have been possible without PCR. A second really interesting use of PCR was made by Linda Buck when she was working in Richard Axel's lab, and she and her advisor Richard Axel had hypotheses that the genes that encoded the olfactory receptors in the nose, that they might be members of a particular protein family called G protein coupled receptors. And there were a bunch of G protein coupled receptors that were known. The reason for thinking that the olfactory receptors might be like this related to biochemical studies that had been done, but really not much was known about the genes. This was in a time before the genome had been sequenced, and you really had to have a fairly hypothesis-driven way to pursue a particular gene if you were after it. What Linda Buck did is she designed a bunch of primers that, in theory, should identify lots of different kinds of G protein coupled receptors, and she put in, bases that would vary at positions that were known to be different in different members of that protein family. And by doing a bunch of these PCR reactions using these degenerate primers, and using the olfactory epithelium as the source of the template that she studied, she amplified a bunch of products. But then the key thing that she did after that was she then digested those PCR products with restriction endonucleases. And the really striking thing is, if you look at most of these bands, the PCR product compared to the digested version, you might see a few sub-bands being produced when you digest. But overall, from what was really a single band or not too many bands, you get some bands that are shorter but not too many. But there was one lane here, lane 13, where she had a pretty strong product with the PCR product that she got. Then when she digested that with the restriction endonuclease, she got tons of small little bands, many more than would add up to be the size of a single product in the original PCR product. So this gave a good clue that there were probably many different genes hiding in that particular band that she had PCR'd. And because she had used degenerate primers that had different varying bases in them, she had in fact amplified a whole family of related genes. And indeed, those turned out to be the olfactory receptor genes. And so you have, many, many dozens of them as the basis for being able to tell different odorants apart in your nose. And this strategy was a way that that very large but related gene family were identified, using the clue that they might actually be G protein coupled receptors. So this to me stands out as a great, clever use of PCR followed by additional analysis of the products that you get using it to make this major discovery of the olfactory receptor genes. Okay, now we move to the part that's getting to be something that's an area of huge interest nowadays, which is sequencing of nucleic acids. The history of attempting to discover the detailed sequence of a nucleic acid, it has many examples of sort of laborious and painful experimental methods. The very earliest complete nucleotide sequence, or complete genome, if you want to think of it as that, was this one of the bacteriophage MS2, which was an RNA bacteriophage. And this was done by this group with Walter Fierce and many of his other collaborators in Belgium and East Barrett. And in these days, there were nucleases that you could use to cut up. This was in 1976 this paper was published, and there have been a number of papers leading up to this one. It was known that there were nucleases that you could use to cut RNA at particular sequences, I think after Gs typically. And if you did that and you got a set of smaller oligonucleotide products, you could then use very painful chemical assay methods and 2D separation as a way to determine what the sequence of those little sub-oligonucleotides were. Then by studying enough of those, you could actually then try to reassemble what the entire sequence had been. And so this was, pushing this kind of methodology as pretty much as hard as you could. This bacteriophage has a genome that's about 3,000 bases long or so, 3,500. And using this method, they were actually able to sequence the whole thing. So this stands out as a really difficult task, but it was the first example of sequencing the whole genome of an organism, if you call a bacteriophage an organism. In terms of DNA sequencing, the early methods that were applied were not too different in some ways from that, although a little more efficient. The one I'll talk about here is the Maxim-Gilbert sequencing. So this method uses chemical cleavage at particular base positions followed by gel separation of fragments. And so the way that this works is you basically took your DNA piece, you labeled it with a a radioactively labeled phosphorus, typically with a kinase kind of reaction. And you then treated the DNA in ways that would favor the breakage of the DNA with a later chemical treatment at particular base positions. So, for example, there was a reaction condition where I think it was depurination of both guanine and adenine nucleotides that would actually result in cleavage at either G or A. There was a separate reaction involving methylation of guanine that would result in cleavage at G preferentially. A third reaction was one that would target the cytosine and thymine, either one. And a fourth reaction would be cytosine only. So you can tell that with these four combinations of reactions, you would get a set of fragments that would end in either G or A, or just G, C or T, or just C. And if you did that, then you would then be able to reconstruct what the sequence must have been, because you'd have a fragment of a particular size. For example, this one here, this fragment was produced in both the G plus A cleavage condition and the G only cleavage position, therefore it must be a G. And so using this sort of an approach of cleaving at particular base positions followed by separation of the products with the label allowing you to visualize the bands, you could actually reconstruct the entire DNA sequence. So this was used quite a bit. However, it was eventually overtaken by another method that was developed by Fred Sanger, shown here. And this is a method, Sanger was instrumental in developing many of the sequencing methods. I haven't talked about protein sequencing, but that was also something that he really was a leader in and had won a Nobel Prize for as well, prior to his work on DNA. So the Sanger strategy, which became very successful and widely used, was one where you hybridize a primer to the sequence that you want to obtain. So this is the template you want to identify the sequence of. You hybridize a primer to it, and the primer is now able to be extended by a polymerase if you have a mixture of dideoxynucleotide triphosphates. And the nice innovation that Sanger had was, if you include in the mixture of your DNTPs some dideoxynucleotide triphosphates, which are ones that will stop the reaction from extending further, because not only do they lack a 2' hydroxyl, like all DNA molecules would, and each of their deoxyribose sugars, but the dideoxy ones, they also lack the 3' hydroxyl. So once you add that nucleotide on, you can no longer extend the chain, it just stops there. So the idea is that you set up 4 different reactions, one with dideoxy ATP, one with dideoxy TTP, one with dideoxy GTP and CTP, and you also have you have these in sort of limiting concentrations, and otherwise there's just a mixture of the regular 4 dinucleotide triphosphates. If you then allow this extension reaction to happen in each of those separate tubes, what you get is, in this tube, the strands are all going to end at an A base position. So they go until one of these dideoxy ATPs is incorporated, then it stops. And similarly with T, GC. And in the more modern versions of this, you've actually also included a fluorescent label that lets you see the A as one color, the T as one color, the G as a third, and the C as a fourth color. But you don't have to do that. You could actually just label the primer with radioactive nucleotide and then run the product separately in wells of a gel. And if you do that, again, because the products of a given length from a particular reaction will end in that base, you know that any strand that is this particular that is in this lane ends in a T. So you know that in this case that position in the sequence was a T, the next position to it was a G, 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 T, etc. Here I've shown them with the colors, but you wouldn't necessarily have to have fluorophores on them. You could just have them radioactively labeled and just have the black band that you would detect with by putting a photographic film against the gel once you've run the gel out. The advantage of using fluorescently labeled dideoxy terminator bases is that you can then run all the products in the same lane effectively, or in something like a glass capillary, which will separate them by size, and you can still tell them apart. So this is a more efficient way of doing the same reaction. But this method, where you're synthesizing a new strand, but it stops at particular positions based on the way that you set up these dideoxy terminator reactions, this led to a huge number of discoveries And for example, this is the method that was used in the form of the somewhat automated ABI sequencing instruments. This was the way that the first human genome was sequenced, and the way that many of the human genes that have been identified were first identified. Now, in the period since then, there's been a wave of new technological developments that have actually produced instruments that are a lot more efficient than the Sanger sequencing method. But a lot of times the Sanger method is still gone back to as sort of the gold standard, where everyone understands the performance of that kind of method quite well, and it's used as a way to validate the results of these new instruments that can produce way more sequences for a given amount of expense or in a given period of time. So that leads us to the present day, where high-throughput DNA sequencing, or these generation of new instruments, have vastly decreased the cost of isolating DNA sequence information. And biologists are fond of drawing these plots where Moore's law looks like a fairly shallow line, which up until now had been very impressive in terms of the number of calculating elements you can have on a particular silicon wafer, for example, or similar things for the amount of storage that you can have in a particular device. The amount of sequencing that you can get for a given dollar amount has gone down by orders of magnitude just within the span of 5 years, basically, or 5 to 10 years at most. And this has completely transformed the kinds of experiments that you can do easily in biology now. And they have big medical implications. So I'll just give a little bit of background about these new technologies and how they work before giving some other examples where high-throughput DNA sequencing has been used to do interesting things. So overall, if we look at these high-throughput methods compared to the Sanger and Maxim Gilbert ones that I talked about previously, in these methods, by and large, you're no longer having to measure the length of a strand. Instead, you're actually isolating your templates and separating them spatially from each other, doing a sequencing, typically sequencing by synthesis, although I'll also mention there are some other methods that use hybridization, and then detecting in each cycle of synthesis which bases have been incorporated. So this sort of a method, since you're using a camera to take a picture of many, many different reaction wells at once, it allows you to capture much more data in a given period of time. And so these are basically very miniaturized reaction chambers where the sequencing by synthesis reaction is happening. And I'll describe in a little bit of more detail about how you can actually tell what base was added. Most of the methods that have achieved wide use so far are ones where you're not truly sequencing from a single molecule of template. Most of them involve some sort of an amplification step, which I show here. The two methods that are that are best described in this slide are the one from the 454, which is now owned by Roche, and Ion Torrent, owned by, Life Technologies. Each of these have some similarities, but I'll describe the differences. So you start off with your DNA that you want to sequence. You make it very dilute in a solution, and you make an emulsion of the aqueous phase of that solution in the presence of these captured beads and with an oil phase that's going to basically enclose little aqueous droplets. These aqueous droplets, in effect, are like little mini PCR tubes where PCR can go on. The capture beads have an oligonucleotide attached to them that will capture one strand of your template. Typically, you've actually attached linkers to your DNA before you actually put it into this process. Using these emulsions as little mini PCR chambers, you can actually carry out millions of PCR reactions at once in a regular size laboratory tube, an Eppendorf tube. And so you can, if you have the other components needed for PCR, including the enzymes, the dinucleotide phosphates, the buffer conditions appropriate, and primers for the other end of this PCR reaction, what you can do is amplify your single molecule of template that went into this aqueous bubble in the emulsion and produce a bead that's got lots of amplified copies of the template stuck to it. That's helpful because then you can break the emulsion, you can put the beads into little wells in a fabricated plate, and this is where the sequencing reaction will take place. So now you've got these amplified templates, they're all from the same molecule, hopefully, stuck to a particular bead, and you can have a camera that's imaging down on top of this plate and it's going to see what happens in each of the wells. Now the sequencing by synthesis methods, They involve something similar to the Sanger method that I talked about, where you start off with a template you're going to sequence, you have a primer hybridized to it, you have a polymerase that's about to extend that new strand. And in these two different methods, the ion torrent versus the 454, you use a different way to detect whether T, A, G, or C have been incorporated in each cycle of sequencing. So the way that the 454 and ion torrent work is you do cycles where you have really only one of the bases present, so DTTP, and would be, for example, one cycle. And you monitor each of the wells in this flow cell where you have your templates that are hooked up ready to be extended. If T is the correct next base to be added to this primer, in other words, if A was on the opposite strand, then you'll actually see that that base got incorporated. The way that you can tell that happened in the 454 instrument is that you have a series of coupled enzymatic reactions that when the deoxytrinucleotide triphosphate is added, pyrophosphate is cleaved off as part of the normal reaction for that addition. And pyrophosphate can be used to generate ribosyl ATP, which is the a molecule that's frequently used to drive enzymatic reactions. That ATP can be used to power the luciferase enzyme to oxidize luciferin, which is a small molecule substrate, and give rise to an emitted photon of light. So the bottom line here is that if T is the correct next base in this particular product being sequenced, in other words, if A is the correct complementary strand base, then what you'll see is a flash of light from the plate from that well during that cycle. If T is not the correct next base, then you don't see any light coming out. And if there's 3 T's in a row that should be added here, what you get is basically 3 times as much light coming out of that well. So you just have a camera that records that, and that can tell you what the sequence was at that position. In the case of the ion torrent method, it's similar in spirit in some ways, except that it uses a very different detection method. So you have similarly cycles where you're having only T, or only A, only C, only G. And in each of those cycles you're monitoring to see whether the trinucleotide triphosphates are incorporated. The deoxynucleotide triphosphates. In the case of the ion torrent method, what you're monitoring is whether protons are released, because that's another thing that happens when you incorporate a new base into the DNA strand that's growing, or a new nucleotide. A proton is effectively released, and the bottom of the wells in the ion torrent instrument are effectively miniaturized pH meters that can tell you whether a proton was released in that cycle of incorporation or not. What that lets you do is detect by a purely microfabricated method, similar to what's used in the silicon chip industry, to detect whether there was sort of a complete solid-state detection system, detect whether or not a base was incorporated in that cycle. And then, so in a similar fashion, if 3 times as many T's were added because there were 3 T's in a row, and then in the correct synthesis, or in the correct next 3 positions, then you would see that much more a proton amount being released, and that would be able to be monitored by the electronic detection system below this. So these methods have become widely used, and enable you to get millions of sequences from a given source in a single instrument run, and for a cost that is now from mere hundreds of dollars up to maybe $5,000 or $6,000 per instrument run. So that's a really amazing advance. The next instrument method I'm going to talk about is Illumina, and just very briefly, this also uses a sequencing by synthesis approach. It's different from the 454 and ion torrent in that you don't use emulsion PCR to amplify your templates and then spatially separate them by putting the beads that have captured those products into the wells of a micro plate. In the Illumina method, you dilute your template and then you allow it to flow onto a flow cell. The flow cell itself has capture oligonucleotides that are going to hybridize to one of the ends of the DNA template that you put in there. And it's a very elegant system, because what you can do is, once you've captured your template strands, you can then carry out PCR on the surface of the plate, and through cycles where the primer is extended, and now it's basically still attached to the plate like it was initially. Those products then can hybridize to other primers that match the other end nearby. And if you can do these cycles many times, you'll get spatially localized clusters if you've diluted your template amount correctly in the first place, stuck to the flow cell, ready to be sequenced. And so then in this instrument, the next step is, again, a sequencing by synthesis sort of approach. And what happens is the DNA polymerase is ready to add on the next correct base in this sequencing cycle. Unlike in the 454 and ion torrent, you don't just have one kind of base at a time being added. In this case, you have all 4 different ones, but they're each labeled with a different fluorophore. And that fluorophore also acts basically to prevent extension beyond a single base in each cycle. So that works very nicely. You, in any given cycle of the sequencer, you incorporate the new labeled bases. You look down at the spots on the flow cell where each of these little amplified colonies is sitting, and you look and see what color is there, and that tells you what base was incorporated in that template in that cycle. And so if you repeat this many times, you can sequence, a good length of the sequence there. And currently, the 454 sequencer, it can read up to somewhere around 500 bases at least nowadays, and the Illumina sequencer is still a little bit shorter in terms of a single read length. 250 is the current number. But on the other hand, there's also ways that one can do paired end sequencing, so sequence from both ends of a given template molecule, and that can effectively give you around 500 bases from the same template. So this is the Illumina method, and overall this has captured more market share and is sort of the workhorse method for most of the genome centers that are doing a lot of high-throughput sequencing nowadays. A final strategy that I'll mention, and this is using one example. There's a company, Genomic Health. They have their own method for doing the sequencing, and they've chosen not to sell instruments to other people, but to have a sort of a centralized facility where they do the sequencing. But it uses a different principle, one that's somewhat similar to one that's in a another instrument category, which is the solid instrument. In this case, the sequencing is not sequencing by synthesis. It's actually sequencing by hybridization of oligos, and I'll explain that in a minute. Like the other methods I talked about, this one is not sort of truly sequencing a single molecule of template at a time. What it's doing is an initial template amplification step, and there's a fairly complicated way that the initial library is set up for sequencing in this approach. Suffice it to say that you, by a combination of doing targeted ligations of the DNA that you want to sequence, and then using restriction enzymes that cut a certain distance away from a known linker sequence in the linker that you attach to the DNA, you generate a sequence of tags, which I'm labeling here as colored segments, and you end up with a circular molecule that's got lots of these little tags that you're going to sequence subsequently. These templates in this scheme are amplified by rolling circle amplification, which is a method adapted from some bacteriophage polymerase approach. You crank out a really long linear copy of this circular template, and the linkers between the areas that you're going to sequence have been designed so that the DNA will bunch up afterwards and form what this genomic health company calls DNA nanoballs. They then have a flow cell arrangement where there are patches where the DNA nanoballs will settle down and stay in a particular fixed location. So now you have lots of amplified copies of the tags you're going to sequence. And the way this sequencing method works is you have oligonucleotides that are degenerated at each position except for one particular position. And here I'm showing a simplified version of this, but the fixed position, you know what base is there based on the color of the fluorophore that's attached to the oligonucleotide. So basically everything else is, NNN, so it could be any of the 4 bases except for this one fixed position. Now what you then do is, in your unknown template that you want to sequence, you hybridize this mixture of oligos, and you have an anchor primer that's in an area of sequence that you do know. If the base that you're querying, or the one where you know what the identity of the base is in that position, if it's the correct one, then then it'll hybridize stably to this template that you're sequencing. You can then ligate those two together and it'll hold on more firmly. And from that, by doing multiple cycles where you vary the position that's known and carry out this sort of hybridization followed by ligation, you can then infer what sequence was present in the unknown tag region of that template. And so in a similar manner as I described with the Illumina, the readout is by looking at what fluorescent color is present at each spot during each cycle. And that's basically how that instrumentation works. So I forgot to give the parameters, but basically the Illumina sequencing platforms at present can generate hundreds of millions of sequences of the read lengths that I specified, like somewhere around 150 bases of paired end sequencing, or now up to 250 bases in the latest version of kits for a slightly smaller version of their instrument they have. This is a vast amount of sequencing capability, and the cost is really quite low. So it's made it feasible to sequence human genomes for not much more than a few $1,000s right now. And similarly, there's been an intense competition between these companies, so the prices have been driven down significantly. Now I'm just going to talk a little bit about some applications of this technology to some interesting areas of biology. This is a paper that came out a while ago, 2005, but I think it was a great example of how you could do experiments that would just be not as easy at all without the power of the high-throughput sequencing methods. This one was done with a 454 instrument, but the basic story was that this group had identified a new antibiotic that seemed to kill Mycobacterium tuberculosis quite well. And they wanted to figure out what is the target of the drug inside the cell, what proteins or what other areas of the cell is it attacking. And they used a very nice strategy where they selected for bacteria that were resistant to the drug. They did this in multiple independent experiments, and they also used several different mycobacterial species and did similar experiments. Then what they did is they sequenced the genomes of all these bacteria that had developed resistance and compared them to the strains that they were derived from. So there they were looking for, well, what genes have become mutated that resulted in these cells becoming resistant to the drug. And then to narrow down, well, which of these mutated genes could actually be the target of the drug, they then compared the independent isolates where they had done this selection experiment to see if they have had any mutated genes in common. And so here's the drug, and they basically they found when they compared these different strains, which were Mycobacterium smegmatis, to the tuberculosis ones, that really there was only one gene where they found that there was a common mutation in those different strains, and was identified more than one time. And that was in the, as you'll see from the title of the paper, it was in a component of the ATP synthase, which was a key metabolic enzyme, and it turned out that it was actually the it was the F0 component of the ATP synthase in the bacteria that was mutated in all the independent strains that they studied, and so that gave a reasonably good hypothesis that was actually the target of the drug in this case. That's something that you couldn't imagine doing that experiment without this vastly scaled up DNA sequencing capacity, and that certainly leads in the direction of being able to use this sort of method for a lot more similar kind of brute force experiments where you isolate an interesting biology in cells and you want to correlate it with gene variants. What I'll not talk about are just some other examples using high-throughput DNA sequencing technology and some of the lessons that have been learned so far. One of the most striking papers in this area was the first example of a human cancer genome being sequenced and then being compared to the germline DNA from that same individual who had the leukemia in this case. This is work from Richard Wilson, Timothy Lay, and Elaine Mardis and colleagues at Washington University. And the experiment here was, they picked a kind of acute myeloid leukemia that seemed like a reasonable bet for one that might have other mutations in it. So they picked one that had a normal set of genetics so it didn't have any obvious translocations or large lesions like that. And they also picked one where there were some internal controls because it had what are known to be recurrent point mutations in AML in the FLT3 gene and in the NPM1 gene. They used the Illumina sequencing platform of the time, and this paper dates from 2008. So with the Illumina sequencer, they obtained 33-fold coverage of the leukemic cells, 14-fold coverage of skin biopsy fibroblasts from the same patient. And then they evaluated those too. They compared them, and they compared them also to other genomes that have been published in the literature. And so, just to give you a scale of the number of variants that one finds in this kind of an experiment, if you just look in general at what are the number of sequence variants that are detected in the tumor sample compared to any given reference genome, they found around 3.8 million. And they used a process of filtering to isolate, or to focusing on ones that were better supported by the sequencing data in terms of the depth they had and so on. Now, interestingly, of these 2.6 million variants that were reasonably well supported by the evidence, there were 2.58 million of them were also present in the skin of that patient. So that shows that the reference genomes that they were being compared to were just different for that person compared to the other genomes that were available. So by far, the majority of these sequence variants were not things that were unique to the tumor. They were actually just part of that person's own genome. From that, there were these 63,000 tumor-specific sequence variants that were identified. Of those, about half of them were also found in dbSNP, which is a database of single nucleotide polymorphisms that has been called, or out of the various sequencing projects that have gone on since the Human Genome Project, and also in the single genomes of Watson and Inventor that have been done in the meantime. Of those 30,000 remaining new single nucleotide variants, they chose not to further examine 20,000 or so of those because they were outside of gene regions where you could readily interpret what they might be doing. Of the 11,000 that were in genic regions, there were some that were in untranslated regions, some that were intronic. Of the ones that were in coding sequences, there were 241, so it's already getting narrowed down quite a bit. 60 of those would be predicted not to change the coding sequence, so they were not really evaluated much further. So that left 181, and of those variants, these were the ones that were predicted to alter gene function, so they were going to change an amino acid or change a splice junction. And 7 of those were unable to be validated when they tried to test and see if they could prove that they were there again. 152 of the 181 turned out to be also present in the wild-type sample when they resequenced. So they were just false positives. They didn't pick up in the tumor, they didn't pick up in the germline sample from the skin that those variants were also present there. And they found 14 were validated as germline single nucleotide variants, and 8 were validated as somatic single nucleotide variants, or these are ones that were, you truly had good evidence that they were probably acquired mutations in the leukemia. So then, naturally, you would want to know, well, okay, what are those 8 genes, and how often are they seen in other leukemias, for example? And the sort of, well, I don't know, potentially surprising, potentially disappointing result was that of all the genes that they found that were the candidates for being new somatic mutations, they sequenced 187 other AML cases and didn't see any of those mutations in any of them. So, what to do? Well, the answer to that was to sequence another leukemia genome, and in this case they did a second AML case, again with normal cytogenetics. This one had an NPM1 mutation. Oh, and I should have mentioned that in the first paper they did identify the FLT3 and NPM1 mutations, so it wasn't that they were necessarily missing a huge number of mutations, at least by that limited evaluation. In the second genome that was sequenced, the case turned out to have a similar coverage, although they got a little bit deeper coverage of the fibroblasts and a little less of the leukemia in that case. in this case, they did find some recurrent mutations, so they identified with a similar filtering scheme to the one I described. They found that there were several different mutations, but in particular this one, IDH1, or isocitrate dehydrogenase 1, they found that it was present in 16 of the 187 other AML cases that they looked at. The other ones that they identified, they didn't see in any of the other cancers they looked at. And this was very striking because that was a gene, IDH1, that had been seen in some glioblastoma multiforme brain tumors and rare cases of colon cancer. So there was some evidence that it might be a cancer-associated mutation. And this thing has triggered a lot of renewed interest in the metabolism of cancers, because isocitrate dehydrogenase is one of the enzymes in the Krebs cycle, and you could wonder whether it would be responsible for a way that cancer cells might respond to growing in hypoxic conditions or growing under conditions where the normal highly oxygenated environment may not be present. That's a large topic that I can't get into here. Subsequent follow-up about IDH1 mutation has shown that it appears to be a negative prognostic marker in AMLs with normal karyotype that lack FLT3 or NPM1 mutations. And I think that this sort of was the opening bell of what's probably going to be a long period of molecular subclassification of cancers up ahead, and something that's going on as we speak with great intensity. Many different categories of cancer are being sequenced by large consortiums organized in all over the world. And more recent papers have identified this IDH1 gene as also one that's being found to be mutated in myelodysplastic syndrome, including the refractory anemia with excess blasts and refractory cytopenias with multilineage dysplasia categories, and also in chronic myelomonocytic leukemias. So it seems to be broader than just an acute myeloid leukemia mutation. So that's sort of just an initial glimpse of the cancer genome sequencing area. Another striking paper was one that came out in 2011, and hairy cell leukemia is a B-cell malignancy that for a long time really didn't have any recurrent or characteristic mutations associated with it, unlike a lot of the other B-cell lymphomas like follicular lymphoma, where you have IgH rearranging with the BCL2 locus and causing a fusion product there, or mantle cell lymphoma, where you have cyclin D1 being rearranged to be under the control of immunoglobulin enhancers. So the hairy cell leukemia was a bit of an outsider in not having a recurrent mutation associated with it. This group here, they decided to do sequencing of not the whole genome, but of the exome of a patient and their germline control, and look for mutations in hairy cell leukemia. So in that one case, they identified a mutation in the BRAF gene of a particular kind, BRAF 600E, and they then looked at 47 other cases of hairy cell leukemia, and lo and behold, they saw the same mutation in every single other case. They also evaluated a bunch of other B-cell lymphomas and didn't ever see that mutation. So this was greeted with great excitement, and it was thought to be quite a good advance that now, okay, there is a hallmark molecular lesion to be associated with this hairy cell leukemia. And I'm sure they must have been delighted when they did those 47 other cases, or evaluated them for this lesion. Subsequent work by some other groups has largely supported these results. This is just showing that they have some pictures in that paper from the cases that they evaluated, and they show that they really have markers characteristic of hairy cell leukemia, and the hairy cell leukemia case had the mutation, whereas this other splenic lymphoma that was of an uncertain category did not. Some other investigators have followed up on this story and have found that by and large, in almost all cases, or certainly in the vast majority of hairy cell leukemia cases, you do see these BRAF mutations. And BRAF is a recurrent oncogene that you see in lots of other cancers as well. So that is borne out. But these other investigators, they did find that around 1/5 of the classic hairy cell leukemia cases, based on the immunophenotype, did not have the BRAF mutation. So not as black and white a result as the original paper had indicated, which is, of course, a common theme in the medical literature. Often the first paper on a topic is, shows the most striking or dramatic results, and then other papers sort of give more, I guess, discover that may not be the case absolutely all the time. This other paper also did verify that in cases that were not the clear-cut classical hairy-cell leukemia, they did not find BRAF mutations. So overall, it seems like those results are holding up well, and that's a great insight that's come from the use of high-throughput DNA sequencing to study this particular kind of cancer. And I think, certainly for people training in pathology and having to learn the details of all these different malignancies, as well as for anyone interested in this whole area of medicine or science, this is just the beginning of what's, I'm sure, going to be a vast area of new knowledge, probably one that will require extensive time and effort to correlate with how well did the different tumors respond to different kinds of treatments. It's probably something that can't just be done in an observational fashion by treating everybody as you would anyway, and then later sequencing them to see what mutations they had. There probably needs to be incorporation of these kind of methods into randomized trials so that you actually truly have a randomization step to make sure that you're not somehow biasing the treatment that people receive in a way that could disguise the true correlations between genotype and response to treatment. But it's certain to be a very interesting time ahead, and one that should be of a big benefit to patients, one would hope. So in summary for this talk, I tried to give an overview of the way that technological advances in the methods for studying DNA and RNA have led to many of the major biological discoveries in the past century, and especially in the last 50 years. And largely as a result of the high-throughput DNA sequencing methods, we were in the midst of one of the greatest expansions of human knowledge about genetics ever. And this is something that I think will bode well for ability to improve our understanding of the genetics related to patient disease risks, to the actual pathophysiology of the diseases themselves, and to be able to understand the responses to treatment better eventually as well. But this is not going to be a 5-year project. It's going to be something that will take undoubtedly many decades to really unfold, and there will be, I'm sure, many surprises along the way as well. So thank you for your attention. This is, as I mentioned, the first in a series of 10 lectures from Stanford Pathology Department and other colleagues to give an introduction to the area of clinical genomics. And I should also mention we've made effort to obtain copyright permissions for the images used in these talks, and I've gone to a lot of trouble to do so. But if you notice some image that you have copyright to, and you feel like it hasn't been properly, permission has not been given appropriately, please contact us, and we'll be happy to rectify that situation. All right, thank you very much, and I hope you enjoy the rest of the lectures.